Experiments done by William Tavalga provide evidence that fish have pain and fear responses. For instance, in Tavalga's experiments, toadfish grunted when electrically shocked, and over time they came to grunt at the mere sight of an electrode. In 2003, Scottish scientists at the University of Edinburgh and the Roslin Institute concluded that rainbow trout exhibit behaviors often associated with pain in other animals. Bee venom and acetic acid injected into the lips resulted in fish rocking their bodies and rubbing their lips along the sides and floors of their tanks, which the researchers concluded were attempts to relieve pain, similar to what mammals would do. Neurons fired in a pattern resembling human neuronal patterns. Professor James D. Rose of the University of Wyoming claimed the study was flawed since it did not provide proof that fish possess conscious awareness, particularly a kind of awareness that is meaningfully like ours. Rose argues that since fish brains are so different from human brains, fish are probably not conscious in the manner humans are, so that reactions similar to human reactions to pain instead have other causes. Rose had published a study a year earlier arguing that fish cannot feel pain because their brains lack a neocortex. However, animal behaviorist Temple Grandin argues that fish could still have consciousness without a neocortex because different species can use different brain structures and systems to handle the same functions. Animal welfare advocates raise concerns about the possible suffering of fish caused by angling. Some countries, such as Germany, have banned specific types of fishing, and the British RSPCA now formally prosecutes individuals who are cruel to fish. Most fish move by alternately contracting paired sets of muscles on either side of the backbone. These contractions form S-shaped curves that move down the body. As each curve reaches the back fin, backward force is applied to the water, and in conjunction with the fins, moves the fish forward. The fish's fins function like an airplane's flaps. Fins also increase the tail's surface area, increasing speed. The streamlined body of the fish decreases the amount of friction from the water. Since body tissue is denser than water, fish must compensate for the difference, or they will sink. Many bony fish have an internal organ called a swim bladder that adjusts their buoyancy through manipulation of gases. Although most fish are exclusively ectothermic, there are exceptions. The only known bony fishes, infraclass Teleostae, that exhibit endothermy are in the suborder Scombroidea, which includes the billfishes, tunas, and the butterfly kingfish, a basal species of mackerel, and also the opa. The opa, a lampriform, was demonstrated in 2015 to utilize whole-body endothermy, generating heat with its swimming muscles to warm its body, while countercurrent exchange, as in respiration, minimizes heat loss. It is able to actively hunt prey such as squid and swim for long distances due to the ability to warm its entire body, including its heart, which is a trait typically found in only mammals and birds, in the form of homeothermy. In the cartilaginous fishes, class Chondrichthyes, sharks of the families Lamnidae, porbeagle, mackerel, salmon, and great white sharks, and Allopiidae, thresher sharks, exhibit endothermy. The degree of endothermy varies from the billfishes, which warm only their eyes and brain, to the bluefin tuna and the porbeagle shark, which maintain body temperatures in excess of 20 degrees C, 68 degrees F, above ambient water temperatures. Endothermy, though metabolically costly, is thought to provide advantages such as increased muscle strength, higher rates of central nervous system processing, and higher rates of digestion. Fish reproductive organs include testicles and ovaries. In most species, gonads are paired organs of similar size, which can be partially or totally fused. There may also be a range of secondary organs that increase reproductive fitness. In terms of spermatogonia distribution, the structure of teleosts testes has two types. In the most common, spermatogonia occur all along the seminiferous tubules, while in atherinomorph fish they are confined to the distal portion of these structures. Fish can present cystic or semicystic spermatogenesis in relation to the release phase of germ cells in cysts to the seminiferous tubules lumen. Fish ovaries may be of three types: gymovarian, secondary gymovarian, or cyst ovarian. In the first type, the oocytes are released directly into the chylomic cavity and then enter the ostium, then through the oviduct, and are eliminated. Secondary gymovarian ovaries shed ova into the chylome, from which they go directly into the oviduct. In the third type, the oocytes are conveyed to the exterior through the oviduct. Gymovaries are the primitive condition found in lungfish, sturgeon, and bowfin. Cyst ovaries characterize most teleosts, where the ovary lumen has continuity with the oviduct. Secondary gymovaries are found in salmonids and a few other teleosts. Oogonia development in teleosts fish varies according to the group, and the determination of oogenesis dynamics allows the understanding of maturation and fertilization processes. Changes in the nucleus, ooplasm, and the surrounding layers characterize the oocyte maturation process.